Hola, buenas. Bienvenidos a otra edición del BioInfo Club. Y como hemos anunciado en las redes sociales, este BioInfo Club se hará en inglés, así que voy a empezar. Welcome to another edition of the BioInfo Club. As we announced, okay, we will be talking in English in this BioInfo Club, so we hope to have a more international audience this time. I'm Ivan Elson, and I'm pleased to present you this activity. If you are watching this video in Eventbrite, you can watch it in full screen by clicking the title above the video and going to YouTube. And in YouTube, you can put the full screen mode and you can write your questions in the chat. So today we introduce you to Anne Martinez Larrinaga. She has a bachelor in biochemistry from the University of Navarra. Afterwards, she did a master's in computational methods by the same university. Right now, she's a PhD student at the Josep Carreras Research Center. She's part of the Cellular and System Genomics group and Endothelial Pathobiology and Microenvironment groups. She's working on the development and application of new computational approaches for the analysis and integration of single cell and spatial genomics, interesting, in the context of cancer and immunology. Today, she will be talking about how to use gene expression data in order to improve the treatment of some subtypes of lung cancer. So this sounds like a really interesting topic. So thank you very much for being with us today, Anne. And you can start your presentation whenever you want.
Okay. So thank you for having me today. It's a great pleasure to be here. First of all, before starting, I really wanna thank my tutors or my tutors, Silvestre, Vicente, and Mikel Hernández, because they were the ones who actually let me do this project and like be in charge. So thank them for the guidance. So let's see. So as we know, lung cancer is remains one of the most common cancers worldwide, both in terms of incidence and mortality. Lung cancer patients, they rarely live more than 5 years. So lung cancer can be classified, taking into account histopathological features, in two big groups: small cell lung cancer and non-small cell lung cancer. We focus our analysis in a non-small cell lung cancer in the two main groups that are lung adenocarcinoma and lung squamous carcinoma, because they account to the 85% of the total lung cancers. Just you to know, both LUAD and LUSC, they have a high mutational rate, but if we look in each group, in LUAD, there is 1/3 of the patients, they lack any druggable genomic mutations, so they can benefit from any targeted therapies. So you to know, the other 2/3 of the group has mutations in KRAS, LAK, and so on. But in LUSC, there is no targeted therapies yet because they haven't found any druggable genomic mutation, so they cannot benefit from these kind of therapies. Thus, there is a need to identify subgroups of patients amenable to new pharmacological treatments. The main hypothesis of this project was that we were going to be able to identify different subgroups of LUAD and LUSC based on their gene expression data. Furthermore, we hypothesized that once we got the groups, we were going to be able to understand the molecular and cellular mechanisms that made us have different groups. And with that, we were going to be able to predict specific drugs that may induce an anti-tumor effect on each patient's phenotype. As a result, this project has the objectives of this project were, first of all, to perform a clustering analysis to classify our patients of LUAD and LUSC in different groups attending to the genotype, to define a global biological characterization and description of the different groups obtained at a cellular level, at the molecular level to identify specific transcription factors that could act as master regulators of each group, and last but not least, to predict candidate drugs for the potential treatment of each patient group through a drug repurposing analysis based on gene expression data. So before going on, I wanna explain a little bit the workflow, because I think at the end it's the most interesting part of the whole project. So first of all, we download the data from the DGCA, the RNA-seq data. We pre-process the data. And I have to add that in LUAD, we got rid of these patients that have common mutations in oncodrivers. These patients that are able to benefit from targeted therapies, we got rid of them because we were not interested in analyzing them. And in LUSC, we took all the patients. We pre-processed the data. We did a clustering analysis. Spoiler alert, we identified 3 different groups in both pathologies. After the clustering, we did a differential expression analysis in order to get the signature of each of the groups. Once we got the signature, was like the base to perform the rest of the analysis. With the signature, first, as I explained, we did a biological descriptions of the clusters. As you see, we did different analyses. I'm not going to explain all of them because some of them we didn't get the results we wanted or we were expecting. So from these ones, I'm going to explain the enrichment analysis, the immune microenvironment analysis, and the survival analysis. Then we perform a gene regulatory network. In this case, the package I decided to use, it's been developed by the department I was working in. So it's still developing, and you could only make comparisons one by one. So that's why we compare cluster one vs cluster 2 and 3, and so on. And last but not least, we did a drug repurposing analysis where you just had to classify your signatures with the genes upregulated and downregulated, and there is like an online tool that is able to make the predictions you are looking for. So for the clustering, the clustering, it took us 4 months because there were so many issues, computational issues, theoretical issues, different kind of issues, but we were able to get the groups, obviously. The algorithms we used were non-negative matrix factorization and k-means, both stochastic algorithms. We decided to use non-negative matrix factorization, NMF, because nowadays it's really common to use it and it gets really robust results. And in k-means, we use k-means because it's a really basic algorithm, and the basic idea of NMF, generally speaking, is based in k-means. So the data we use, the count matrix, like the input of the algorithm is just use the matrix, the count matrix, where you have samples in columns and features in rows. And the k values we try were from 2 until 7. So the results were the following ones. In NMF, what we used to actually know, the output of the clustering, it's a consensus clustering. A consensus clustering is a kind of graph where you have samples by samples, and each dot or each square of the sample of the graph represents the probability of these 2 samples grouping together or clustering together in each iteration. So as you can see, we are looking for, as the name says, a clustering that is consensus, that is robust, that is not messy. This what does it mean? That we're gonna work with k equal 3, k equal 5, as you can see in both cases, because when it was k equal 6, 7, it was less robust, or the probabilities were most messy. So this data suggested us to work with k equal 3 and k equal 5. In k-means, we decided to use 3 indexes. Each of them it values or it measures different things. But at the end, we have like the same suggestions that k=3 and k=5 were the best ones. However, in some cases, k=4 was actually equal also fit. We decided to work with k=3 because, first of all, when k was bigger, bigger groups were split in smaller groups, and there was we didn't find no biological relevance or no bibliographical evidence to work with k=5. Also, we trusted more the results from NMF than from k-means. So yes, in LUAD, okay, NMF has been used in different studies, and they have shown that the results we obtain are more robust. Also, in these studies, and taking into consideration our results, regardless of the genomic profile you are using, this means that if you consider the genomic mutations or you don't consider them, and you use NMF and the gene expression data from the DGCA, you obtain 3 robust subgroups in LUAD. But if you use other type of algorithms or other type of data, the number of groups vary. These tell us that actually when you are working with clustering algorithms, the decision of the algorithm and the data set to be used may affect the final result. In LUSC, no one has used NMF, and there is no, but there is only one study that they did like an analysis really similar to ours, but they find 4 groups. You may ask, okay, they find 4, you find 3, what's going on? The thing is that there is a discrepancy between how their workflow and my workflow. So it could be actually explained because of that, like the way they pre-process the data and the way they obtain the clusters is a bit different from mine. So that could, and also they are using a microarray data set instead of publicly available data. But it is important to mention that the World Health Organization recognized only 4 subtypes of LUSC: basaloid, small, clear, and papillary. But basaloid and small are considered to be like the same one. So this kind of like this seems to align with my data or with my results. Okay, once we have our groups, as I said, we obtain like the signature of each of them to do like a biological description of what we have. So what we did to obtain the biological description, we did a functional enrichment analysis. As you know, there are 2 types of analysis. We can do an over-representative analysis or a gene set enrichment analysis. The main difference between both of them is that in a gene set enrichment analysis, you consider the gene expression levels, like you actually, the input, it's the log fold change of your signature. So this actually leads to discover changes in the expression of genes that share a same categorical class. This is like a summary of we actually obtained because there were so many graphs. So just in a few words, you see that we have an up and down regulation of the immune system in both pathologies, but that it's like 2 groups that they have like opposite expression profiles. And then we have an upregulation of cell proliferation processes in the remaining group. So afterwards, what we decided was to study the tumor microenvironment because previous groups have studied using bulk RNA-seq. So why we decided to study it? Because at the end, the tumor microenvironment, it has complex interaction between various type of cells and could be determined in the patient's prognosis. And as I said, we had an upregulation of the immune system, and we wanted like to characterize or to discover why this was happening. So for this, we did 2 approaches, as we have been doing in all the analysis, also to compare the performance between the different tools. So we use CyberShard that gets you the proportion of tumor immune infiltrating cells. And we use an approach from a paper published by Faruqi, where in this paper they use the cell signatures from other paper, and with that cell signatures they obtain the log fold change for each of the genes, and they do like the median of the genes. At the end, what they got is like a heat map with the expression levels of different immune cells. So what we obtained was in LUAD, we didn't get any kind of, it was not in the survival, it was not significant because the samples, we had a few samples, they were only like 61. But it was interesting that we see that in group 1, there was an upregulation of common immune checkpoints. Okay. Then in LUSC, it was more interesting because we did find differences in the survival of our groups. So we were like, okay, what is causing these differences in the survival? Like, why we see this? So when we got like the heat map with the expression levels of the different cell types, we see that the group 2 that actually has like one has a worse survival than the other 2 groups had there was an up and down regulation of B cells. So at the end, our conclusion, or in summary, we can say that the tumor microenvironment and the proportion of tumor immune infiltrating cells could play a role in the prognosis, and in this case could actually be also could play a role in the identification of the subgroups. Okay, at the next level, what we decided to do was to so we all know that transcription factors may play a key role in cancer progression. So we hypothesized that we could use them as good markers, like as biomarkers of our groups. So what we did was we did a gene regulatory network to identify rewired transcription factors. Rewired transcription factors are transcription factors that they present differences between different conditions. Apart from that, from the ones that were rewired, we also look for those that were rewired but also differentially expressed. The gene regulatory network was based on a model-based approach because because in one article they said that the model-based approach is more accurate, it produces less positive, and the rate between true positive and true negatives is maintained. So this is what we got. Like in summary, these were like the transcription factors we suggest as possible biomarkers for each of the groups. You can see that there is no transcription factors in group 2 because there were some computational issues, and I don't know why. They didn't, like the script didn't run and didn't want to work. So it was interesting because we found that in group 1, there was a downregulation and upregulation of transcription factors related with cell proliferation processes. But in this one also. But then it was curious because there were transcription factors that were common between groups, but with opposite expression profiles, like SOX7 and SOX2. In one group was downregulated and in another one was upregulated. And we actually saw this also in the gene set enrichment analysis. And last but not least, we found out that TP63 is a really common transcription factor that is usually upregulated in squamous cancers in general. Okay, so the last step was we have found our groups, we did our biological description, we did the molecular, like the cellular and biological description of the groups, we found the transcription factors, and just the last step was to actually like predict potentially effective pharmacological treatment for both groups based on their signature. To do this, we did a transfer proportion analysis. There are different approaches that you can follow. We decided to do a signature-based computational approach due to the data we were using. These kind of approaches are really, they heavily rely on public accessible gene expression data. So we decided to use the connectivity map platform. So in this platform, it's a project that is based and is, it's been done by the Broad Institute, that they like develop a new sequencing technique. So it was cheaper to actually sequence. So they have so many data and about about different cellular states. So it's a really cool tool. In the connectivity map, to do the drug reproportion analysis, they use cellular responses to perturbations to find relationship between diseases, genes, and therapeutics. So what we find was that we focus like in the top 10 drugs that would predict. It was curious, it was interesting because each of the groups, they had different drugs were predict, which may also indicate that we did actually have different groups. Also, the drugs that were predicted are related with the biological description of each of the group. For example, we can see that in LUAD group 2, it has been proposed VEGFR inhibitors. Just you to remember, in this group, there was an upregulation of cell proliferation processes. The transcription factors were also related with angiogenesis and these kind of processes. It has been seen that VEGFs, it's immunosuppressive, like that the immune response, it's not significant. You won't remember, but when we were analyzing the tumor microenvironment in this group, there was like the immune system was downregulated. When I say downregulated, it's like that doesn't mean that it was like I had less cells. It's just like the expression of the genes that characterize those cells was low. So we hypothesized that in this group, if you use VEGFR inhibitors, you could actually improve effectiveness of immunotherapy and enhance the immune system. And in LUSC group 2, one of the examples was that in group 2, we have also the PI3K inhibitors. As you all know, the signaling pathway of PI3K AKT enhance a really immunosuppressive phenotype. It's really PI3K is really upregulated in a lot of cancers, and it has different effects. So in this group, we saw that there was like an upregulation of common immune checkpoints and a low presence of T cells. So we hypothesized the same as in the other example, that if we inhibit the survivability of the tumor using PI3K inhibitors, we could promote the response of other immune cell types and maybe get better results. So just to summarize really briefly, what like the conclusion of this project was, first of all, NMF and K-means as clustering algorithms using TQCA data identify 3 different patient subgroups in both LUAD and LUSC. However, a non-negative matrix factorization is a more robust algorithm. Then, in terms of functional enrichment analysis, the groups presented different biological profiles. 2 groups had opposite expression profiles in terms of immune system, and the remaining one was characterized by enrichment in cell proliferation processes. Then, in LUAD and LUSC, the 3 groups had different tumor microenvironment composition, which determine differences in the progression and prognosis of lung cancer. Then, most patient groups presented unique transcription factors that were not only differentially expressed, but also rewired. Last but not least, potential and unique drugs that could reverse the group signature were proposed. As with the transcription factors, the proposed drugs were also related with the biological profile of the groups. So this was all. I know it was not much, but the project was like really long, so I tried to summarize as much as possible. I hope you have liked it. I really enjoyed it, and I think that we were able to take really cool results at the end.
Okay, thank you very much for the wonderful talk. It was very interesting. Personally, I can see similarities with my current PhD project, so it was especially enriching for me. And now we can start the question time. We can wait. Wait, we have a question. Okay. So Have you thought about doing enrichment analysis of transcription factors?
It was like the following step of the project, but I didn't have enough time, and it was really, so the packages I was using as it was being developed, it had a lot of issues, and it was really picky on how you had to put the data and everything. And as I was working with really huge matrices, the computational cost was like, I don't know, a week to obtain a list of 10, 20 transcription factors. So I ran out of time, and yes, I got the transcription factor. But the following step was to see, like, to do the enrichment with the transcription factors, but I wasn't able to do it.
Okay. So we'll wait some time for any more questions. Okay. Another one. Why did you choose k-means as the other clustering algorithm?
As I said, k-means is known by all of us. We know that it always converges, so that was like a positive thing about it. It was really easy to implement, and it was really easy to understand what's going on in compared with NMF, that you have to understand, like, that you are using meta-genes and that you have two matrices, like a matrix that then you become two matrices. It was more complex. K-means was more easier. So we use it as we wanted to compare both clustering algorithms performance, and NMF, like the main basic, generally speaking, or really basic speaking, the idea behind NMF, it's like based in k-means. So that's why, like, I was speaking with my tutors, and they were like, oh, this is really similar to k-means. So we are gonna see k-means and see how it's like, what happens between both algorithms.
Okay, nice. So, well, remember we have a survey you can fill out. We have another question first. Did you try to find if your clusters were present in the Cell Encyclopedia?
We actually tried it, but so we downloaded the RSG data from the Cell Encyclopedia, and we tried to do the same clustering analysis and see if we got three groups. So we used the signature of our group to actually cluster the cells, but we didn't see any clear pattern or anything. So that was like the following step, if the project will follow on, was to actually see, like, validate what our groups that actually exist on cell lines.
Nice. So in the meantime, once again, we have another one. What about comorbidity with other cancers?
What?
Comorbidity, the association with other cancers.
We didn't focus in other cancers. We just studied lung cancer. Like, I don't know.
Yeah. Okay. Similarity with other cancer interactions. Similarity. Yeah. No. You didn't focus.
Study it. No.
Okay. Yeah.
No. It was just a year-round project, so we didn't have much time to actually compare with other kind of cancers.
Okay. Why did you have challenges when you ran into clustering algorithm?
It was fun. First of all, it took me some time to actually understand what I was supposed to do. Then I had to figure out how to validate, because as is stochastic nature, you have like to actually validate in some way with different subsamples that you are actually getting k = 3 or k = 5. So that's first of all. Then it took so long to run it on the cluster, like 10 days, because of the validation process I was following. And then I was a bit full, a little bit dumb, and I was running the matrices without knowing which column was each patient. So I finished the analysis and everything, and I didn't know which patient was each. So I had to do it all over again, but knowing which patient was each patient. So that's why it took me so long. I had like a lot of inconvenience, one after the other one.
Yeah, classic mistake. It happened to me too. Okay. So once again, thank you, Anne, for being with us this afternoon. And we hope to have you here again in the future, so you can tell us more about your research. Also, thanks to the attendees for coming and for their interesting questions. And last but not least, I want to thank our collaborators, Larrinaga, PTS Granada, and MedinBio for supporting us. I'm gonna share the screen. Okay. So as many of you already know, we are BioInfoGRX, an association that works to share bioinformatic knowledge originated in Granada, although we reach people from everywhere as we work mainly online. We organize talks and workshops from basic to advanced bioinformatics. You all can follow us in our social media for updates, so you don't miss any event. And if you are interested in collaborating with us with some talk or some workshop, feel free to contact us. As I said, we have posted a link to a survey in the chat, so you can tell us if you liked this activity and keep improving our content. Remember that this video will be recorded and posted in our website, bioinfogrx.es, along with the slides and additional information. And I also wanted to remind you that we have our next event en la jornada de bioinformática de la UGR. Ya lo voy a decir en español porque el siguiente evento va a ser en español, que será en la jornada de bioinformática de la UGR el día 25. Así que estad atentos. Y nada, muchas gracias, Anne. Y nada, creo que no se queda ninguna pregunta más. No. Ok. So, thank you very much. Muchas gracias y nos vemos en la siguiente. Bye.